Welcome to Biology Made Easy. Today we are looking at structural organization in animals, specifically chordates, and we are looking at structures in cavities. We'll look at the practical of this, looking at a dissection, and we'll look at toad, guinea pig, and albino rats. Structures are not haphazardly placed in the organism. Structures are compartmentalized in cavities, and the cavities, I have put it together in two major categories known as external cavities and internal cavities, also known as body cavities. Description: external cavities are cavities that open to the outside, can be seen by the naked eye. Internal cavities or body cavities are known as coelom. They are fluid-filled cavities within the body. They house internal organs. So we will first of all deal with the external cavities, and examples of external cavities: you have buccal cavities, nasal cavity, eye socket, ear canals. Here I have the lower jaw of a guinea pig, and here is the skull of a guinea pig. It's a mammal. Piecing it together, I have here as the buccal cavity. Here is the buccal cavity. And then we have the nasal cavities. See the holes in there are the nasal cavities. And I have the eye socket. It's a cavity in which the eye lies. Then I have these holes. This is one of the hole here and one here on each side of the skull. These are the ear canals that the ear organs are in. These are the external cavities. The origin of these cavities, we take the origin from the embryo, and in our previous lessons, we saw that the embryo has these major layers of tissue. We have the ectoderm, mesoderm, and coelomates have a coelom, that's a fluid-filled cavity. Then we have the mesoderm, and then we have endoderm and the gut. So the external cavities, the origin is the gut. They all open to the outside of the body. Good. Now we want to forget the external cavities and concentrate on the internal cavities, also known as body cavities. The body cavities, they are within the body. They house internal organs. What is the origin of the body cavities? The origin of the internal cavities, that's the body cavities, is the coelom, right? Now, this is a coelomate. We have the ectoderm, mesoderm, and has this fluid-filled space, coelom, and then you have mesoderm, endoderm, gut. Now, the internal organs are formed by the mesoderm, and they lie in this fluid-filled cavity, which is the coelom. And to see the body cavities, we need to open up the organism. But let's use this as an example. I have this. Let's imagine that skin covers this structure. If I remove the skin, we have a body wall. That's the structure of chordates. Remove the skin, you have the ventral body wall. Then you have the dorsal body wall. If you open up the body walls, between the ventral and the dorsal body wall is a space known as the body cavity, which is fluid-filled. And the organization is such that this fluid-filled cavity, organs lie in them. And the organs that lie in them, we will soon see that they are organs that were formed from the mesoderm. So here we are with our ventral body wall and dorsal body wall, and the skin is removed. We will get the body cavity, and the body cavities house internal organs. So with this illustration, we have this mammal. This is the skin. This is the ventral body wall. These are cavities. We want to look at examples of the cavities that we'll see. The ventral surface, we will see the thoracic cavity. we'll see the abdominopelvic cavity, and from the dorsal surface, see the cranial cavity and the spinal cavity from the dorsal body wall. So now we want to use practical dissection to look at these cavities and their contents. Look at the body cavities that are in the ventral surface, thoracic and abdominopelvic cavities. We need to cut, and so we have a toad here, amphibian. That's the ventral surface. Make a slit in the middle there, and here is the skin, and here is the ventral body wall. So here is the ventral body wall of the toad. Here is the skin removed. Here is the ventral body wall of an albino rat. It's a mammal. Here is the skin removed. So here is the body cavity of the toad. When the ventral body wall is opened, these are the structures we find in it. We have the stomach, we have the heart here, here's the gallbladder, liver, intestines. This structure is the ovary with eggs in it, and we have fat bodies. You can see the fat bodies well here. These are fat bodies. Here is the stomach. Here is mesentery holding the stomach onto blood vessels, etc. And we want to look at the thin wall, so we brought all these three. Here is the heart, here is the liver and intestine. This is what is in the abdominopelvic cavity of the toad. There are other structures beneath what we are seeing now, which we will see, all in the abdominopelvic cavity. Here we have the ventral wall of the guinea pig opened, and we see the cavity. Here is the abdominopelvic cavity in situ. Now, in the abdominopelvic cavity, we see the stomach, liver, intestine, large intestine, small intestine. Then we see the urinary bladder, see the testes. Here we have an albino rat. We have the ventral body wall. Here is it removed. Look at this space. That is the body cavity in which these are the digestive system. We have the stomach, etc. here. And then we have part of the reproductive system. This is a male. This structure is called uterus masculinus. All of them in the abdominopelvic cavity. And here is the thoracic cavity. Here is the xiphoid cartilage. Now, look at the difference between the thoracic cavity and the abdominopelvic cavity of the mammal. They are separated. Here is the thoracic cavity. Here is the abdominopelvic cavity of the mammal. In the amphibian, the toad, the thoracic cavity and the abdominopelvic cavity is one large cavity. There's no diaphragm that separates it. This has a diaphragm. Here is a xiphoid cartilage, the diaphragm here. Here, there's no diaphragm to separate these two cavities there. Abdominal cavity from the thoracic cavity. So here is the heart, clearly seen when you open up the ventral body wall of the amphibian. So in the amphibians, that's the toad, you have the one large cavity, thoracic cavity, heart, lungs here, abdominopelvic cavity, forming one large, let's take note. Still in the abdominopelvic cavity, if we push the digestive organs, we will see that there are more structures that can be listed that are in the abdominopelvic cavity. So the push here, we see the uterus masculinus, we will see the urinary bladder. Here is the urinary bladder in the guinea pig that we can see very well. We can also see this vast difference. We can see the epididymis, all of them in the abdominopelvic cavity. Here are the two kidneys with their blood vessels. Here is a uterus. Also, all of them in the abdominopelvic cavity. Now, I want to look at the contents of the thoracic cavity. When the thoracic cavity is cut, the ribs here are cut, and the thoracic cavity opened. This is what we see. Here are ribs cut. Here is part of the ribs not cut. Here is the xiphoid cartilage. Here is the heart. Lungs are here. Here's a big blood vessel. Here is aorta, and the vessels here, we'll have time to talk about them. But this is the thoracic cavity opened. So here we also have this one. This is the thoracic cavity still. Here has the diaphragm. It's turned over to see the diaphragm, which you couldn't see here. So you have the diaphragm, you have the heart, and you have the lungs in the thoracic cavity with the blood vessels. Now we also want to note that the heart has a membrane around it, and the membrane encloses a fluid. We have a membrane very close to it, and this membrane is a pericardial membrane. There's a fluid in it, and it's pericardial fluid. Then the lungs also has membrane around. The membrane is pleural membrane, encloses a fluid, pleural fluid. So the pericardial fluid and the pleural fluid, their function is to prevent friction as the heart contracts and relaxes, and as the lungs expands and relaxes, there's no friction. But then that makes it that within the thoracic cavity, there's a cavity for the heart and also a cavity for the lungs. So you have a thoracic cavity and cavities inside cavities. That's the thoracic cavity. So we have discussed the ventral surface and the cavities that could be seen: thoracic cavity, abdominopelvic cavity. Now we look at the dorsal surface and look at the cavities that we would see. Now on the dorsal surface, we'll see the cranial cavity and the spinal cavity. The cranial cavity is in the head, so I have the skull of the guinea pig, and here you can see a hole in it, and that hole is the cranial cavity. And then I have a vertebra here. Here is the atlas. You see the hole inside it? This is the hole that the spinal cord lies. And this hole, you have other vertebrae joined end to end. It forms the spinal column, and that forms the spinal cavity in which lies the spinal cord. And this can be seen on the dorsal surface. So here, the skin, the dorsal body wall, we want to look at it in our toad and then albino rat. And this toad, that's the dissection of the toad. Here is the dorsal body wall. Here is part of the vertebral column that has a hole, that's the spinal cavity in which the spinal cord lies. This is the, so here is also the section through the albino rat. This is the dorsal body wall. Look at all the many structures attached on the dorsal body wall. And is this dorsal body wall, if we look at it from behind, we will see the vertebral column that has a space, and that forms the spinal cavity in which lies the spinal cord. So this ends our journey through cavities in chordates. If the lesson has been useful, please give us your thumbs and kindly do subscribe if you haven't subscribed already. Goodbye.